DNA viruses. Again, good afternoon. So, for the pox viruses, there are many under this category. So, characteristically, they are linear, double-stranded DNA virus. They have a brick-shaped envelope, and the size of the genome is approximately about 130 to 300. So, they are rather among the larger viruses. Okay, next, classification of the pox viruses. We have three groups: the orthopox virus, the capripox virus, and the suipox virus. So under orthopox virus, we have variola or smallpox, vaccinia, and monkeypox virus. For capripox, we have sheep and goat pox viruses, and swinepox virus for suipox virus. So the genome we have mentioned earlier is linear, double-stranded, and they have specific sequences for gene regulation. They also have genes for viral proteins and for enzymes that are needed for replication. Life cycle, so the virus enters the host cell, usually the epithelial cells of the lungs, okay, via direct fusion or by endocytosis. They replicate in the cytoplasm, so this is the first of the virus we have encountered so far that are capable of viral replication in the cytoplasm. So usually they do that in the inside the nucleus. So there are two transcription stages. It can be the early transcription and late transcription stages. After transcription, so the virus, okay, the new viruses can now be released by the cell, okay, either by cell lysis or by budding. The first among the members of the pox viruses is this variola virus or smallpox virus. So the condition is called smallpox. This is a highly contagious disease, and transmission is by droplets, respiratory droplets, or by direct contact with lesions. So signs and symptoms, there may be fever, malaise, and the presence of skin lesions similar to monkeypox. Pathogenesis of variola virus. So the kind of cell, as I mentioned earlier, is epithelial cells of the respiratory tract. So the spread of the virus will start from the skin, okay, to the internal organs until it reaches the circulatory system. So the rashes are characteristically pustular, so meaning that it contains pus, and it may result in severe complications afterwards. Vaccinia, the next one, is used to produce the smallpox vaccine. Okay, it has a similarity to variola, however, it's less virulent. So what else? So, well, we have mentioned that it was used to produce the smallpox virus. Monkeypox virus, so it causes monkeypox. It is a zoonotic disease, so meaning it is transmitted from animal to man or even from human to human by direct contact. So by eating meat of wild animals, you may get this monkeypox infection. Clinical manifestation is fever, lymphadenopathy, and rash that is similar to smallpox. So other pox viruses include cowpox. So that is also used. It was also used to develop smallpox vaccine. Okay, and it can lead to a mild human disease. Orf virus. This is the one that is infects sheep, and this is common among people, those, or especially the farmers, those or the shepherds, those who handle sheep, especially for shearing, or that is to remove their wool. Clinical implications. So there used to be smallpox, but now it's already successfully eradicated because of vaccination. So this was in 1980. However, right now we have ongoing threats, of emerging cases of monkeypox and other zoonotic pox viruses. So maybe we should look into the practice of eating wild animals, because they may pose a great threat to spreading these kinds of infections, zoonotic infections. So how do we diagnose? We can use PCR, and we can use serological testing by identifying antibodies against the virus. And additionally, we can use histopath. By doing biopsy, it can reveal the presence of the inclusion bodies that will allow us to identify the infection. So management, there is now vaccine available, which is derived from vaccinia. And in case you are exposed, we're talking about monkeypox this time, because there is no more smallpox. So post-exposure prophylaxis or vaccination can be done if you are exposed to someone who is known to suffer from monkeypox. Supportive care is also given symptomatic. So we address the symptom. If there is fever, you give paracetamol. If you have, other signs and symptoms, you address those problems. Prevention is by vaccination. And we know that there are other ways to, like what we did during the time of COVID. So we can utilize surveillance and containment, or that means quarantine, okay, in case of outbreaks. So for research, there are research areas that may be used to investigate more about this virus by looking into the genetic diversity and evolution of pox viruses, and by looking into the potential therapeutic uses of vaccinia virus to treat cancer. Oncolytic pox viruses. So there is such a thing as oncolytic pox viruses. So they are engineered or technologically developed viruses that are used to target, replicate, and kill cancer cells. So far, there are clinical trials that have shown the safety and efficacy of this treatment to different types of cancer. So the role of pox viruses in gene therapy. So pox viral vectors can be used to deliver therapeutic genes. So they are like carriers. So they are used as carriers. And because viruses are usually target specific, so they can find the abnormal genes that can be being targeted. So, however, there are challenges because there is immune response against a vector that can limit the effectiveness. So that means the body can still recognize this as something that should not be present, and therefore it might attack. It might attack the vector if used as carrier, or so the carrier molecule. Epidemiology, prevalence is high. There is high seropositivity for vaccinia because of exposure to the vaccine. Seasonality, so this is quite similar to seasonality of some viral infections, so usually during summertime, so especially in the tropics. Immunological response, so there is natural immunity and vaccine-induced immunity against pox infections. This is important. Next one, challenges. Bioterrorism, the threat of bioterrorism. Even if it's already eradicated, so the Centers for Disease Control still keeps a copy of this virus in their headquarters, so in the laboratory. So we need to be prepared. We have to stockpile vaccines, so make it available for future use, and develop rapid diagnostic tests in order to identify people easily who are suffering from these viral infections. Public health implications, so monitoring and education. So these are very important techniques, okay, or strategies uh, to check on how many people, or who are the people who are infected, and that people will be aware of how it is transmitted and other important matters. So emerging pox viruses. So there are now new variants, okay, and these newly identified viruses are being studied different research um, activities. Potential risks, so we need to assess their implications for human health. Community awareness campaigns, so people should be made aware of how to prevent the transmission and how to prevent infections. So maybe one of the ways to deliver viruses is by school-based education. Future directions of vaccine development, so investigate new vaccine formulations that may be more effective than the previous ones. Gene therapy, explore new applications, although right now this kind of technology is not really very available in the developing countries like the Philippines. Alright, so let's have anelloviruses. So they are small, circular, single-stranded DNA viruses. They don't have an envelope, and their capsid is icosahedral in shape. So they are very small, measuring only about 2 to 3 kb, so the genome. So that means that the DNA is very small. Classification, we have the following: torquetenovirus or TTV, torquetenominivirus or TTMV, and torquetenovirus 2 or TTV2. Components of the genome or the DNA, so we have the circular single-stranded DNA, and they contain the ORF or open reading frames, so that's the meaning of ORF, okay, that encodes for viral proteins. Replication, usually it requires the host cells for replication, definitely. So how do they cause infection? So they enter the host cells by receptors. What else? They replicate inside the nucleus, okay, and then they rely on the whole cell machinery for synthesis of the DNA. Release, the virions will then be released by a cell lysis or by exocytosis. So this is a little different from the previous virus. It's a torquetenovirus or TTV. So far, this is the most studied member of this family of viruses. It was first identified in 1997 in a patient with post-transfusion hepatitis. So clinically, it is found in patients with liver disease, but its role is not really very clear. Potential effects, there may be immune modulation that is associated with various diseases, and there is high prevalence in patients who are immunocompromised. Transmission, so it may be acquired by blood or body fluids, possibly by respiratory secretions and by sexual contact. So this is commonly found in healthy individuals, but most adults are seropositive. Clinical implications. So since they are associated with especially liver diseases, so we need to investigate. Okay, so if a patient has liver disease, they may be transmitting not just hepatitis viruses but also anelloviruses. And we know that immunocompromised patients are easily infected with this type of virus because of the weakened immune systems. How do we detect? So there is a PCR technique for detecting the TTV DNA in serum and other body fluids, and serologically, so there may already be serological tests to identify antibodies. Okay, so since this is not really very common, so hospitals may not necessarily be requesting them, so this may be considered as some sort of special test that might require the hospital laboratory to send out their specimen. Management. So currently there are no antiviral treatment available for infections. Management strategies should focus on symptomatic treatment. So just like in other viral infection, we should address the symptoms rather than the disease. Okay, so if there are liver implications, so that naturally we have to focus on directing those symptoms. So regular follow-ups are necessary for immunocompromised patients. In patients with hepatitis, okay, so we don't really know if this is related to uh, the severity of the condition. However, so yeah, we might as well investigate whether okay this super infection with TTV can influence the severity of liver disease. Research. So we can there may be a possibility of exploring the exact role of TTV in liver disease, as we have mentioned, and investigate potential associations with other diseases. Evolution. So another thing that may be studied will be related to genetic diversity and mutation in the viral particle. Okay, immunological response to anellovirus, there is immune evasion. So that means that this virus may persist in the cell without causing the disease, so they are asymptomatic, so undetectable. Antibody response, so the immune response in controlling viral role, okay, to this infection. So since this is undetectable, so the body may not be able to produce corresponding antibodies against the virus. Prevention, so since there is no vaccine, what we can do is to practice standard precaution, universal precaution in preventing transmission of body fluids, okay, infection in the healthcare setting, the hospital, in clinics, in laboratories. Okay, comparative genomics, so they have compared this with other viruses, okay, to find whether there is a relationship. Okay, importance, this is to help identify the potential pathogenic variants that is present among the viruses. In blood transfusion, other implication. So this is also found in blood donors. Okay, since this is not being actively screened, so there is a chance that this may be transmitted by blood. Okay, but since there is no known disease associated with this condition, so this is uh, still a threat, but not necessarily a problem. Ongoing studies to assess the risk and develop screening guidelines. So maybe in the future, this will be one of the viruses that will be screened for during if someone is going to donate blood. Case studies. So maybe a group of scientists may review significant TB infections and their management and learn, okay, the implications for clinical practice and public health responses. Okay, so there are new variants of this virus, so therefore they may also be monitored for possibility of, more pathogenic strains. Therapeutic applications, so again, there may be a possibility that this can play a role in gene therapy. Awareness, so education, so people should be made aware that there is this anellovirus that can be transmitted, okay, by various means. Okay, surveillance, so this is another public health implication, so we need to check for possible TTV infections in immunocompromised patients. Challenges, so since this is not really a mainstream topic, mainstream subject in research, people may not actually be interested in doing research about TTV. Okay, data collection is challenging because, well, we are not really familiar with TTV infection. So it just coincides with uh, hepatic disease. Data collection for data collection. Impacts of co-infection. So we should focus on the co-infections because we know that when we can compare what for people who are infected only with hepatitis and those who have co-infection with TTV. Relevance, so it has its implication in patient management strategies. So how do we deal with patients who are suffering not just from hepatitis but also from TTV? Regulatory aspects, so policy development, so by developing guidelines to monitor anellovirus in the health setting, and efforts to increase awareness and understanding about this type of infection. Polyomavirus. So this is a family of small non-enveloped DNA virus with circular double-stranded DNA, icosahedral capsid structure, and the genome measures only 5 to 7 kb. Okay, so there are many genera, but the most notable are polyomavirus, that includes the human polyomavirus, and simian virus 40 or SB40. So for the genome, they have the early genes and the late genes responsible for viral replication, transformation. The last one is for encoding for proteins. So replication relies on the host cell machinery. So that means that the host cell will be the one to manufacture the DNA. So they enter the host cell receptors by receptor. So that means this is usually the cell membrane. Then they replicate inside the nucleus. Okay, and what else? So to release the new virions, it may lead to cell lysis. So the key human polyomaviruses are the JC virus and the BK virus. So they are associated respectively with PML, progressive multifocal leukoencephalopathy for JC virus, and for BK virus associated with kidney disease, like those who have received transplants. Pathogenesis. So they infect oligodendrocytes in the nervous system, the CNS, that leads to demyelination, and other neurological symptoms. So individuals who are at risk are immunocompromised, people, especially those who have HIV or AIDS. Pathogenesis, usually they reactivate in people who are immunocompromised, and this is also associated with people who have received renal transplant but failed. Symptoms of infection are hematuria, flank pain, so side pain, and graft dysfunction. So maybe the virus itself has a contribution to the failure of the graft. Transmission, so they are spread by urine and respiratory secretion. So they are also linked to poliovirus, especially the SB40. Prevalence is widespread in the general population. So PML and JCV, so we already noted that there may be neurological symptoms associated with JCV infection, and for kidney transplant patients, it's also the same for those who have BK virus infection. How do we detect these viruses? So it still is by a PCR. Okay, there is no serological test or histological tests that are available for their detection. Sorry, there is a serological test, but it may not necessarily be available, okay, all the time. So imaging. So radiologically, we can perform MRI, magnetic resonance imaging, to diagnose PML or multifocal leukoencephalopathy. Management, there is no antiviral therapy for these viruses. So what else? We should focus on immunocompromised patients because they are likely to be infected by this virus. Reduce immunosuppressive therapy if necessary because that may lead to them being at higher risk for infection. And regular monitoring of viral loads in transplant patients. So there is a technique to measure the viral load. Prevalence is widespread as we have noted, okay, and the risk factors is being immunocompromised. Focus area for research, so the role of these viruses in other diseases, so whether they contribute positively or negatively, okay, in other diseases. Because these viruses are not routinely being detected, so they will only be detected once signs and symptoms have appeared. Exploring therapeutic applications, so possibly you can use this for gene therapy and for cancer treatment. Okay. Epstein-Barr virus and polyoma virus. So we have mentioned Epstein-Barr in previous presentations. So they found out, we should try to understand how co-infection with EBV and polyoma virus can affect the disease outcome. Will it become more severe or will it be lesser? So immune response, the interplay between immune responses to different viruses. So but we have to investigate whether the combination of viruses during infection can affect the severity of the condition, or maybe, yeah, so something similar to that. 16, slide 16, immunological response to polyoma virus. So there is also immune evasion, so that means they remain undetected by the immune system. So because, they are present without causing any disease. So the immune system is also responsible for producing antibodies, and since it's unable to detect, okay, the infection, so the antibody response may be delayed. Lack of antiviral therapies, this is one of the challenges that we face. So you will notice that most of the time, since we follow this kind of pattern in our presentation, so many of the viruses, as of yet, we don't have treatments available. So we have to screen people who are at risk, okay, to this kind of infection, like for BK virus transplant patients. Research. So we need to investigate polyoma virus that may pose health risk and understand the epidemiology of the emerging strains. So again, for public health implication, awareness is very important. People should be made aware, recognize the importance of polyoma virus in the health setting, surveillance to monitor outbreaks, and especially in vulnerable populations. So we're done with viruses. Actually, I think I missed one virus, which is yung parvovirus. Anyway, we will look into that next time. Okay? Thank you very much for listening and watching the video. So sorry that I cannot be with you for this session. So you may repeat if there are things that you want to raise. You can ask that through our GC. Thank you. Bye-bye. Okay, so this is the second part of the presentation this time about parvoviruses. So still DNA containing virus. This is the last one. So this is a family of small single-stranded DNA viruses. Okay, they have no envelope, and they have an icosahedral capsid with a very small genome of only about 5 kb. So we have the following genus. Number one is a parvovirus that includes human and animal viruses like the parvovirus B19. And we have the dependovirus, which requires co-infection with a helper virus for replication. So this is not something that is new to us. Genome. So the DNA is single-stranded, and it has two genes, ORF genes, early genes and late genes, responsible for replication and for proteins development, for proteins production. Life cycle is via cell receptors, and replication happens in the nucleus, And what else? It involves synthesis of the complementary DNA because of the virus. So how do we remove or how do we release the particles, the viral particles, by cell lysis? So the first one is parvovirus B19, the most important virus so far under parvovirus. This can cause the so-called fifth disease. So if you remember, there is the sixth disease. Now we have the fifth disease, or erythema infectiosum. So it can lead to complications in immunocompromised people or pregnant women. So pathogenesis is by infecting erythroid progenitor cells in the bone marrow that leads to a aplastic crisis, okay, with anemia. Manifestations are fever, rash, and joint pain in children. Transmission is by respiratory secretions, blood transfusion, and vertical transmission from mother to fetus. So the disease is caused by, or the conditions caused by this would be number one, erythema infectiosum, also known as fifth disease, characterized by slapstick rash in children. Aplastic crisis, so this is similar to aplastic anemia, but people who are at risk are people with sickle cell disease or chronic anemia. For pregnancy, the risk of hydrocephalus or fetal loss. So uh, that means that the fetus, there is intrauterine fetal demise. So that means that the child may die, the fetus may have died inside the womb. Diagnosis is by serology or by PCR, by detecting corresponding antibodies and the DNA in blood or bone marrow. Treatment, supportive care for symptomatic patients, hydration and pain relief. In severe cases, blood transfusion for patients with aplastic crisis. So when the bone marrow is unable to produce blood cells, so there may be a need for blood transfusion. And then, of course, if you are a pet lover, you may have heard of parvovirus in dogs, so canine parvovirus. So this is highly contagious, and this can cause severe gastrointestinal disease. This is when you see, the blood in their stool. So, the other one is feline parvovirus. So, in this case, they can cause panleukopenia. So the WBCs are depressed in cats, and it's also similar to canine parvovirus, so there may also be gastrointestinal disease. Prevalence is widespread, and most people are seropositive. Risk factors, so there may be an outbreak in schools or daycare centers because of children, so, and maybe because of crowding. Focus areas. So we can investigate the role of parvoviruses in diseases beyond erythema infectiosum. So what happens after the child or the person has already been cured of this condition. So what happens next? What else? In exploring therapeutic applications of parvoviruses in gene therapy. So they may also be utilized. There is a possibility that any of those viruses may be used for gene therapy and for cancer treatment. So we are looking not into the bad side, but in the good side of viruses. Currently, there are no vaccines available for parvovirus B19, but for canine parvovirus, there is already a vaccine for them. Natural immunity is available. Okay, and also for vaccine-induced immunity, there are no. So we are talking about human infections. Notable cases of clinical studies, there were reviews on the significant infections in human and animals and the implications for clinical practice and management. Public awareness, so there is a need for education about the disease and its transmission and the challenges that researchers face in diagnosing mild cases, and differentiating them from other illnesses. So that's a challenge, especially for clinicians, for doctors, and for, well, mostly for doctors. So evolution, viral evolution. So they have seen that there are new strains and there's a potential for zoonotic transmission of this virus. Therapeutic uses again, so they are thinking that this may be of use in cancer therapy, and uh, for hygiene, so hand hygiene and avoidance of close contact during outbreaks is suggested. Community awareness to educate the public on the recognition of symptoms so that they may, so by finding, if you find the presence of those uh, signs and symptoms, you may try to report and therefore avoid uh, the spread of this disease. Hygiene, so again, we have already mentioned of hygiene, education, okay. Public health implications, so this is similar to other viruses, surveillance and reporting is necessary. Okay, so, and then focus areas, so pathogenesis, how it infects humans and animals, explore potential links among different infections and parvovirus and co-infections, the effect of parvovirus co-infections with other viruses in terms of severity and outcomes, and clinical relevance implications for patient management and treatment strategies. So, and then finally, future directions in research. So maybe vaccine development for human infections and gene therapy, possibly the use of this virus to provide for gene therapy. So this is the last slide for uh, DNA viruses. So the next time we meet, we will cover RNA viruses, which is more expansive compared to DNA viruses. So thank you for watching. Thank you for listening. Okay, see you next time.